So it's now my pleasure to introduce our first speaker for this afternoon, Dr. Huda Zoghbi. She is a 2017 Canada Gairdner awardee. She's being recognized today for her pioneering work in the discovery of the genetic basis of Rett syndrome and its implications for autism spectrum disorders. She's a professor at the Baylor College of Medicine. She's an investigator at the Howard Hughes Medical Institute, and she's also the founding director of the Jan and Dan Duncan Neurological Research Institute at the Texas Children's Hospital. She received her undergraduate medical training at the American University of Beirut and the Meharry Medical College in Nashville, and then she continued her medical training in pediatrics, neurology, and genetics at the Baylor College of Medicine. Dr. Zoghbi has received numerous awards throughout her career for her accomplishments in the field of neurology, including being elected as a fellow of the American Association for the Advancement of Science, the National Academy of Science, and I'd like to just point out one prize. She received the Jacobs Ladder Prize here in Toronto in the year 2000. This is from the Jacobs Ladder Foundation. It was named after Jacob Schwartz, and it's a charitable foundation whose goal is to improve and understand neurological disorders. Dr. Zoghbi discovered that Rett syndrome, a debilitating neurological disease in children, is caused by mutations in a DNA binding protein, MECP2. She demonstrated in animal models that the brain is exquisitely sensitive to the dosage of this protein, and that the disorder in animal models can be ameliorated specifically using antisense oligos that normalize MECP2 levels. These discoveries now pose considerable potential therapeutic benefit in the treatment of this devastating disease. The title of her presentation is The Story of Rett Syndrome and the insight it provides into neuropsychiatric disorders. Please join me in congratulating and welcome Dr. Huda Zoghbi.
Thank you so much for the kind introduction, and I'm really honored to be in the company of my esteemed colleagues and part of this exciting symposium. So my foray into research was inspired by patients, and this is the first patient that I met who inspired me really to change route from being a clinical child neurologist and get trained in basic science. Ashley was 5 years old when I've seen her, and she was a healthy girl when she was born, did everything a girl will do in the first 2 years of life, including learning how to say words, using her hands, being sociable, even humming nursery rhymes. And all of that changed after her second birthday. And over a period of few years, she lost these abilities and lost the language, couldn't use her hands. You'll see her wringing her hands here, and developed autistic features, balance problems, seizures, and breathing dysfunction. So you could imagine how disheartening it is for someone to go through normal development and to lose these abilities. In the next video, we'll show you some of these activities in motion. You'll see this repetitive hand movement. You will see some rocking activity that's common in autism. And as this young girl tries to walk, you will see also the stiffness in her gait and her inability to really move, although she has the motor strength to move. So having seen quite a few of these girls was very disheartening to me, but at the same time, it really made me wonder, what is it that's going wrong that's affecting them? And that's when I decided to go to the lab and learn how to do genetic research in hope of finding the gene. The challenge was that over a period of my residency and fellowship, and shortly after, I saw about 200 girls, but they were all like this, one in a family. They were all sporadic cases. So in the 80s, it was a big challenge to convince someone that a sporadic disease could be genetics. Nobody really would have considered that. And in fact, people said, work on autism. It's a much more common problem rather than working on Rett syndrome. It's such a rare problem. So it is for that reason that the search for the gene was tough. But because all of these were girls, and to me, the reason I was convinced it was a gene is because of their clinical pictures being so similar. I at least narrowed it down from all the chromosomes to the X chromosome in my search, thinking that we don't see it in males, probably because the male has a single X chromosome, so all of the cells have a bad allele. This male may not make it. Whereas in females, because they still have a healthy X chromosomes in the other half of their cells due to X inactivation, maybe that's why they make it. So that's why we narrowed the search to the X chromosome, and it was really, I'm not going to walk you through all what we did and how we got there, but I'll tell you that 16 years later, after seeing my first patient, we identified the mutation in the X-linked gene, methylcytosine binding protein 2. Now, this protein was first discovered by Adrian Bird, who also received the Gairdner Prize for this discovery. He discovered it as a protein that binds methylated cytosine, and what we discovered is its relationship to neurological function in Rett syndrome, as you can see here in this cartoon, binding methylated cytosine. So today what I'd like to tell you is what have we learned since the gene discovery? What do we learn about neuropsychiatric diseases from studying this rare disease? And what are the effects of the dysfunction of this protein on chromatin and neuronal, on chromatin structure and neuronal function? And I will end on some of the therapeutic approaches we are exploring. So what we've learned is the vast majority, about 95% of classic Rett syndrome cases are caused by mutation in this gene. And here's the clinical phenotypes. And you'll see here that these girls are mosaic. Half of their cells will have the healthy allele and half will have the mutant allele. But what we've also learned that if a girl has a milder mutation, or if she has favorable X inactivation favoring the healthy alleles, they might present mostly with psychiatric features. It may be autism, it may be OCD, anxiety, and sometimes mild intellectual disability. So this first tells us that the vulnerabilities of the brains for different phenotypes is really different. So a milder mutations bring out the more psychiatric and cognitive, whereas to get all the motor and all the other physiologic problems with breathing, then it has to be a more strong mutation. And in males, on the other hand, others and us have seen few males. When the mutation is inactivating, these males cannot live. They will die by the second year of life. However, males with a milder mutation may present with hyperactivity, autism, intellectual disability. Some might present with juvenile onset schizophrenia, some with bipolar. But unfortunately, no matter how mild the mutation is, eventually in their 50s, they might have respiratory dysfunction. So again, showing us the vulnerability of brain circuits that are really important for cognitive and behavioral issues are different from the vulnerability of other circuits. I should mention, since also the discovery of the Rett gene, now we know that over 45% of intellectual disabilities in autism are caused by de novo mutations, and they are mostly sporadic. So I think the idea of sporadic mutations causing disease is now bearing out in other disorders. We could model this disease in mouse models. You'll see here a healthy mouse with the paws apart, and then you'll see the Rett mouse model, one of the Rett mouse model we've generated, this one, where they have the constant ringing of the forepaws reminiscent of the hand ringing in the humans, and they have the tremor that you'll also appreciate. And it is important because there are very few neurologic disorders you can really model very nicely in mice. In this case, the model really recapitulates almost all the features. One surprise came is when we added a copy of the gene. We used the human gene with all of its regulatory elements and nothing else. So it was about a 99 kilobase piece of DNA. When we put that gene in mice, these mice developed a progressive neurologic syndrome, and you'll see that on this video. These mice had one copy of their own endogenous mouse gene, and then the extra human copy we've added. And we then showed that this extra human copy is actually causing the disease simply because we're doubling the level of the protein, not because we added a human gene in the mouse. Because if we put this on a mouse that lacks its own gene, we can rescue all the phenotypes. So it's purely for doubling the protein. So with this information, we and others predicted there have to be humans that might have duplications in this gene. And indeed, many others, especially Professor Vanesh, has found many patients with duplications that span the locus. They are big duplications, and the overlap region spans these genes, but the mouse at least pinpointed the gene as the culprit because the phenotype of the mice and the human is also very similar. This is my first patient with the duplication that I have seen. He came to us because of features of autism, repetitive. You'll see his stereotyped activities going back to his computer, repeating the flapping. And unfortunately, this is a progressive disorder. These males go on to develop a lot of seizures, respiratory problems, and become bedridden. And it is unfortunately a lethal disorder. So on this slide, I summarize what we've learned, that the dosage of this protein matter. If you have none of it, it's lethal immediately in the first 1 or 2 years of life. 50% in every cell or a mild mutations can cause Rett or psychiatric phenotypes. And we discussed the duplication. The triplications, both in mice and human, are even worse. So it's just the same but more severe phenotype. And we know that having too much of the protein causes a gain of function or enhanced function rather than interference with the function, because we did physiology as well as molecular work to show that they're really the inverse of each other. So here are gene expression data from mice totally lacking the protein, and here are the data from our duplication mice. And you'll see the majority of the genes are really just the inverse of each other. What goes down will go up and vice versa. So with this, then we moved on to understand how does the protein affect gene expression. We have a lot of gene expression changes. One of the few things we've done, and many other labs have done that as well, check where does this protein bind in the DNA. And does it bind methylated cytosine? Does it bind to specific regions? And what I will tell you very simply, it binds everywhere there is a methyl cytosine. And whether that cytosine is followed by a guanine or followed by any of the other base pairs, adenine and or cytosine and so on. So the MCH mark is the non-CG mark, and there is a very strong correlation in the binding. And in fact, many of the gene expression changes seem to correlate more with the non-CG methylation. So we know it binds everywhere. We haven't been able to localize it to any specific region. And that was perplexing. We still want to understand how. So to learn the how, we also went back to patient data, particularly males, because we thought the males have a single allele. They don't have the confounding effect of exon activation. And there are many males with different deletions or truncating mutations that have different severities of phenotype. So this is a schematic of the protein. This is the domain that binds methylated DNA. This is a domain that used to be called the transcription repression domain. It interacts with NCOR complex. And you could see here in 2 different mutations, or several mutations, that all of them inactivate or truncate that domain, but clinically they are actually different. And if you look at this table, you'll see a big change between those that have truncation that lead infantile death versus those that live for years. And the demarcation happens just here. A difference of 3 amino acids is a difference between life and death in the first 2 years versus much later on. So this intrigued us, and we thought this might help us at least learn something about the function of the protein or an added function. But there are quite a few humans with the early truncation, but there was only 1 human with this later truncation. So one would worry about modifier effects. So we decided to test this in mice. And we also looked what is exactly there in these truncations. And what you'll see is here's the one that truncates at 273, and here's the one that truncates at 270. And this domain, which is evolutionarily conserved, end up to be a domain that is similar to what's in HMG proteins or AT hook domain that binds AT-rich DNA. And this is typically the AT hook domain, and you'll see this one truncation that's more severe totally disrupts that, whereas the later truncation keeps that domain intact. So we modeled these in mice, and you'll see here again, the later truncation has much better survival compared to the early truncation that looks exactly like null. So this led us to conclude that in addition to the function of the protein being binding methylated cytosines and perhaps mediating some of its effect through this complex, it does have probably an effect on bending DNA or changing the architecture or structure of chromatin through DNA modifications. And we are in the midst now of looking at the 3D chromatin structure using high-C in situ techniques to determine if that contributes to the function of the protein. Part of that is driven by us seeing changes in chromatin. So here is a brain cells from a female mouse. That's a Rett model because they're heterozygous. And you can evaluate the chromatin structure or the intensity of DAPI staining that gives you a readout on how chromatin might be compacted or not in the same animal side by side. And you can appreciate there's more intense DAPI staining in the null. So it's more condensed chromatin or consistent with repression, which is what we see when we do gene expression changes. Whereas if we look at the transgenics, here's a mouse with a duplication, and this particular mouse has three times the level of the protein. And again, we have a side-by-side wild type and triplication cell. And again, you can appreciate the chromatin is different, whereas it's less condensed and more diffuse here and less DAPI. And if we quantify that, you'll see the more DAPI, more condensed chromatin in the null, which is a sign of repression. These animals lack the protein in those cells, whereas in the transgenic, it's actually the opposite. So at every level, we see these two mutants really behaving differently, and this is why we decided to invest in looking at the 3D chromatin structure. Moving to the neurological functions, this circle summarizes all the Rett syndrome phenotypes, and simply put, it affects really every function of the nervous system. So we've decided to study the function of the protein in different cell types to learn if we can attribute some clinical features to particular neurotransmitter systems or neuronal subtypes. And we've done this for many neurons, but I'm going to share something with you by simply looking at the two types of major neurons, the inhibitory neurons and the excitatory neurons. Inhibitory neurons make about 20% of the brain cells. And when we knocked out this protein in inhibitory neurons, we replicated many of the features of Rett syndrome, except for the anxiety and the tremor. And in males, it also caused premature death. And we learned that what happens when you knock out this protein is you partially disable the neurons. In fact, the inhibitory neurons still function. GABA signaling is still there. It's just marginally reduced by 30%, and 30% reduction is enough to cause all these problems, including premature death. This got us interested to then subdivide the inhibitory neurons and study the two most common subtypes, the parvalbumin and somatostatin neurons, work done by these two postdocs, and Kirsten is now at the University of Ottawa, actually, and Aya is back in Japan. And what we've learned from this was also surprising. We've expected to have a lot of overlap in the phenotype, but again, we discovered that while the total GABAergic knockout had many phenotypes, the parvalbumin knockout has more motor, spasticity, apraxia, and learning phenotype, whereas the somatostatin knockout showed the stereotypies and the seizures. So there were modularities within these neurons. However, survival was compromised in both and different ages, but this tells us that all neurons are really important for survival in the network that regulates that in the Rett, but that there is modularity and different neurons have different vulnerability when they're partially disabled. Now, if you totally silence these neurons, you might get a totally different phenotype. But the reason I share this with you, because I think partially disabling a neuron is more representative in what happens in certain neuropsychiatric disorders, because in these disorders, we know all the neurons are there, but they're probably not functioning robustly. Now, when we turn to the excitatory neurons, a work done by a graduate student, we found the features we did not see in the inhibitory knockout, such as the tremors, the anxiety-like behavior. But there were, of course, some shared knockouts. And this is a summary of these data where if you have partial dysfunction of excitatory neurons, anxiety will be one of the first features to see tremor, but eventually they will have problems and they will die prematurely, whereas social behavior deficit, stereotype behavior, and impaired learning is more apparent when partially disabling the inhibitory neurons. And I think what this told us is quite a few important things. That moderate reduction in either GABA or glutamatergic signaling can cause a multitude of neuropsychiatric phenotypes and their slight differences in the vulnerability. And these are severe phenotypes we're seeing in these animals. So I want you to think about this in the context of what might happen in actual psychiatric disorders such as bipolar or schizophrenia or depression or anxiety. There, the reduction might even be much more subtle. And this is probably part of the reason we're having a hard time finding genes that mediate these disorders, because they may be genes that function in the either synthesis or catabolism pathway of these neurotransmitter systems, but it could be very hypomorphic allele, milder allele, and this is what we have to think about. It also told us, and I didn't show the data here when we did the rescue experiments, is that you can't really rescue by just giving modulators of one arm of the system. You have to do both. You have to do probably also serotonin and dopamine. So we're then still challenged. We could not find a solution by doing all these neurotransmitter-specific studies. And with so many genes affected, really the only way to treat this disorder is to find a way to effectively replace the protein completely, either through a gene therapy approach or other, finding a protein that compensates for it. But today I'm going to tell you about an alternative strategies we have taken for both the Rett model and for the duplication model. In the Rett model, we've decided to explore neuromodulation, where you can stimulate a portion of the network, hoping if you focus on a particular phenotype, you can ask the question, can you neuromodulate the Rett brain, and can you bring a benefit to the animal model? And I will then tell you about the strategy we're taking for the duplication mice. So the area of the brain we decided to modulate is the fornix, which has projections to many areas in the brain, but particularly to the hippocampus. And by recording in the hippocampus, you know that if you're stimulating the fornix, you're getting a readout in the hippocampus, and you're not inducing seizure, you're not causing trouble. And this is work done in collaboration with the Tang lab, and Schwang in his lab has done that. And what I'll just summarize the data, it was a really beautiful result in that stimulating the fornix led to the rescue of all the learning, all the hippocampal phenotypes in the Rett model. It also normalized synaptic plasticity. The long-term potentiation became similar to wild-type animal and normalized neurogenesis. So this was quite encouraging and told us that at least this part of the network can be modulated to gain function. And we decided to explore exactly how could that be happening. And this was happening, the exploration, by a graduate student in Stelios' lab, but most of the work was done, the experimental work, by Wei Lu in my lab, where she was looking at the CA1 neurons in the hippocampus and looking at their properties in the various models of MACP2 disorders. And she used a GCAMP transgene so that the neurons, when they're activated, GFP fused to calmodulin will fluoresce. And I hope you can appreciate in the wild-type animal, the few cells firing just spontaneously. There's no, and she recorded to show that these firing coincides with electrophysiological recording. But in the disease models, we've seen a lot more firing, and this is at a time there are no seizures in the animals, and we've gone on to record this in vivo and establish there are no seizures. You'll see a lot more firing together from the neurons in the animal models. And if we were to quantify this, you'll see from multiple neurons, the wild-type cells fire independently, but you'll see, for example, in the Rett female mice, a lot more neurons firing together, and we've noticed this increased synchrony, and we're able to quantify it. And when we asked what does DBS do to this, you will see here that in wild-type animals, whether sham or DBS stimulated, there's not much difference. In the Rett animals, there's this increased synchrony, and then using DBS normalized that synchrony. And there are a lot of details about what may be driving this synchrony, which synapses. I'll be happy to talk to you about that during the reception. But we're beginning to at least dissect the mechanism by which DBS may be working in this area. We've also just found out that DBS can normalize the gene expression for about a quarter of the genes altered in the animals. So from this study, we knew that the Rett brain is, at least in mice, responsive to neuromodulation, and we are now contemplating if we were to translate this into treatment, because for Fornicil DBS can be done in human. So we need to determine the frequency by which this treatment will happen. We would like to know, is other parts of the Rett brain also responsive, particularly the motor function? And we're testing this in other models of intellectual disability, because this should be agnostic of the genetic cause. It should be really more about the network. And eventually a trial could be done, but of course we have to add behavioral therapy to that, because these girls would have been lacking cognitive training for quite a while. As for the duplication syndrome, I think the therapeutic approach is a little bit easier, because we knew first that if we normalize the MACP2 levels in adult mice, we can rescue all their phenotypes. We've done that genetically. Hezi has done that. So the next thing was, can we do something that we will do therapeutically in the clinic? And for this, we collaborated with Ionis Pharmaceutical, and they designed antisense oligonucleotides that can target the human allele and normalize the dosage. By eliminating the human allele from our transgenic mouse, we are left just with the mouse endogenous allele. And you'll see here, for example, here's healthy mice. They're quite active. The transgenic mice, you see them less active. When we give them an ASO, they gain the activity again, and you can see the quantification of their at least behavioral data here. Here's an example of mice that were about 6 to 7 months old. They're having a lot of seizures. We treat them at 6 to 7 months, and we record EEG at the same time. We monitor their seizures. All the EEGs get normalized, and all the seizures disappeared. So this was quite encouraging to us and led us to now deal with the next challenge, which is our mice had 1 mouse copy, 1 human copy, but humans have 2 human copies, and your ASO has to get you from 2x to 1x. If you go less, that's going to give you Rett syndrome. So for that, we created a mouse that have 2 human alleles. One of them is GFP tag under the endogenous promoter, and we've verified that. And the whole point of that is to show if you use ASOs, you can actually titrate the dose of the ASO just like you would titrate a pharmaceutical. And we are now testing these animals to see if we can get therapies, the right dose to then hopefully move that into the clinic. And ASOs have been used in children with spinal muscular atrophy, so we know this is something that we could apply. But because of this discovery and knowing we can modulate the levels of MACP2, we finally decided to embark on a new strategy to find modulators of MACP2 through genetics. And the idea would be we want to find some things that lower MACP2 that we'll use in the duplication, and some things that might increase it, because many of the human patients have mutations that reduce the protein, and if you were to boost the level of that protein, you can help the symptoms. And we created for that, Laura created a transgenic line that monitors MACP2 levels and the health of the cells and transcriptional program of the cells. And then we've designed it so that we can genetically inhibit genes one by one and monitor the level of the protein. And we can find things that might decrease the protein or increase it using this strategy. And here's just one example from such screens. When we use the kinome, you'll see this phosphatase. There's 3 siRNAs that decreases MACP2 levels. And then we independently test short hairpin RNAs where you see MACP2 levels decrease. And this is shown here. And then we go in vivo and test that, and we see the same effect in vivo. And the effect is at the protein level because the RNA is unaltered. We knock down the RNA of the gene, but we see reduction of the protein. So this really opens up now a strategy for us to go screening in an unbiased way the genome. And we started with the druggable genome, and just completed these studies now where we have multiple candidates that regulate the protein. And Steve made these libraries for us. This helps us also learn about the biology of the protein. For example, the enzymes that are involved in its degradation or its modification, all of that will happen from this screen. So with that, I hope I convince you that studying this rare single gene disorders is helping us understand the vulnerabilities of the networks in neuropsychiatric diseases, and that mouse models can be exploited to map the origin of some phenotype, particularly with partial inactivation or dysfunction of neurons. Of course, the brain is sensitive to MECP2 levels, and we hope that through either manipulation of the network or ASOs to come forward with the treatment and maybe even the modulators. Of course, this work was done by wonderful people in the lab, many of whom are faculty members now, and Ruthie is the one who discovered the genes, and many others are working, and the wonderful collaborators we've had throughout the years, and the families who really trusted in us and worked with us and continue to really help us with the research and the funding agencies. Thank you.